Hi everybody, I trust that you are all well and welcome to our lesson on Unit 5. In this lesson we are going to be looking at the topic of animal reproduction and development. We know that reproduction is one of the 7 key life processes that is found in all living organisms, and in this unit we are going to look at the 2 types of reproduction that we find, which is asexual and sexual, and we will also focus on animal development in terms of concepts related to fertilization, embryonic development, and also looking at where the embryo develops and how it receives its nutrients. This is a very saturated topic, so I've chosen to focus on a few important points. So let us jump straight into it. There are 2 kinds of reproduction that occur in the animal kingdom. This includes firstly, sexual reproduction, and secondly, as we will see on our next slide, asexual reproduction. So sexual reproduction has a very important factor attached to it, and that is the concept of fertilization. And fertilization is a fusion that takes place between the haploid nucleus of a sperm cell with the haploid nucleus of an egg cell. And as a result of this, we can form a diploid zygote, which can develop into our new animal. And when we think back to the formation of gametes, we remember that due to concepts like prophase 1 and independent assortment during metaphase 1, we have genetic variation. And so offspring that are produced through sexual reproduction are genetically different. Although genetically similar, they are genetically different as well to their parents. Our second kind of reproduction is asexual reproduction. And unlike sexual reproduction, there is no fertilization that takes place. But we are still looking at reproduction, and so as a result of that, we will have offspring being produced. However, there's just no egg cell and sperm cell involved in this process. There are 4 major types of asexual reproduction. Our very first one is called fission. And fission, F-I-S-S-I-O-N, generally occurs in microorganisms. So although this is looking at animal development, we include this in this discussion as well. And during the process of fission, which is very similar to the concept of mitosis, a bacteria cell or an amoeba cell replicates its DNA, it grows and then splits into 2 new organisms. And we can see that on this image that I'm highlighting in blue. Here we have a unicellular organism, most likely an amoeba, which is growing in the first part, and it is growing so large that it now needs to divide and undergo the process of fission. And to do that, it replicates its DNA, and it then splits its cytoplasm into 2 new offspring. So our first one is called fission. Our second type of asexual reproduction is called budding. And budding generally occurs in organisms present in phylum Cnidaria. If we have a look at the image on the right of this slide, which I will highlight in green, this image shows us the process of budding very nicely. So we see that in the first image, we have a parental organism, and it starts to form an outgrowth. In the second stage, or number 2, that outgrowth starts to undergo some differentiation and specialization so that it starts to look like its parental organism. And in diagram number 3, that outgrowth has grown so large that it starts to now fall off from the parental organism. And in diagram number 4, it falls off completely, and it will then attach onto a substrate, so a rock or a ground, for example. So if I had to draw in a substrate like a rock, it will then attach onto that rock, and we then have a new organism. So that's our second type of asexual reproduction, called budding. Our third type of asexual reproduction is called fragmentation. And a good example of fragmentation is looking at sea stars. So sea stars, as we know, have 5 points or 5 sections in their bodies. And if a certain section gets removed, maybe from a predator or due to a harsh environmental condition, it is possible that firstly, the starfish can grow that section back, but also that section that has fallen off itself can also grow into a new organism. So the reason why it's called fragmentation is because it focuses on the concept or the idea of fragments. And once an organism breaks up into fragments, it can potentially develop into a new organism. Of course, this can't happen in all living organisms. For example, in humans, if your section of a body gets cut off, for example, your hand, it won't necessarily grow back. And if we think about that, humans are vertebrates. And when we've looked at example number 1, 2, and 3, we were focusing mostly on invertebrates. So it's generally invertebrates which undergo asexual reproduction. So that was our third example, and it is called fragmentation. Our last and final example, number 4, is called parthenogenesis. It's quite a mouthful, but simply what that means is that your organism is able to reproduce from an unfertilized egg. And this generally takes place in lizards, and what happens is during the process of meiosis, so remember, meiosis takes your chromosome number from diploid to haploid. But in organisms like a lizard that reproduces asexually through parthenogenesis, that egg cell goes from haploid back to diploid. So it goes diploid, haploid, diploid when we look at gamete formation in organisms that reproduce using parthenogenesis. One of the biggest disadvantages of asexual reproduction is that there is no fertilization. There's no egg and sperm involved. There's no gametes. And remember, gametes are genetically different to their parent, the parental organism that they come from. And because there's no genetic variation in asexual reproduction, this is a large disadvantage for animals that reproduce asexually. So we've seen that organisms can reproduce sexually or asexually. However, once these offspring are present and are within the world, how do the parental organisms ensure the survival of their offspring? That is what we are going to look at next. The first 2 points under this concept says that all species produce more offspring than the environment can handle. And the second point links onto the first one. The proportion that survives is much smaller. We know that a certain environment has a carrying capacity. And once offspring are produced, the population size exceeds this carrying capacity. if we have to define the term carrying capacity, that is simply the maximum number that an environment can account for in terms of the resources available. So when there's more offspring, there's more competition for resources. And as a result of that, only a small proportion of the offspring will survive. And the first 2 points actually link up to the concept of natural selection, which was proposed by Charles Darwin. The small proportion of organisms that survive, these can grow and develop into individuals who will reproduce, and they will thus pass on their genes. We had survival of the fittest, and that is the important aspect that we find within these first 2 points, and why I mentioned that it relates to natural selection. Next, we see that species with external fertilization produce more gametes than species which have internal fertilization. An example of an organism which reproduces using external fertilization is a fish, for example. And an example of internal fertilization can refer to fertilization that takes place in mammals like humans or cows, for example. On the left-hand side, we have an example of external fertilization. Remember, fertilization refers to the fusion of the nucleus of a sperm cell with the fusion to the nucleus of an egg cell. Both of these nuclei are haploid. So during external fertilization, your male fish will produce a large number of sperm cells, which will be released into the water. And your female will produce a large number of egg cells, which will also be released into the water. And a very small fraction of them will be able to meet up and effectively undergo the process of fertilization. A male fish can release up to 10 billion sperm cells, and your female fish can release up to 3 billion eggs. If we now have a look on the right, we are looking at internal fertilization. This diagram shows the reproductive system of a cow, and we have a vagina in this reproductive system into which a penis can enter and sperm can be released. However, there is a protected and set pathway that these sperm cells can follow once inside the cow. And so we find that mammals produce a very small number of sperm cells compared to species that produce externally. So your males generally release about 4 billion sperm cells, and your females release 1 egg every month in a process called ovulation. So if we compare external and internal fertilization, we can see that the reason why more gametes are produced during external fertilization is because there's no direct pathway for the sperm to follow. Once the sperm and the egg are in the water, they can move away from each other more than moving towards each other. And so the larger number ensures that a percentage of them will be able to undergo the process of fertilization effectively. And in this way, we can ensure the survival of the species. Organisms which reproduce using internal fertilization provide greater protection to their embryos and also provide a greater extent of parental care. We then come to the concept of amniotic eggs. And amniotic eggs can be aquatic or terrestrial, but the key idea is that they contain protective layers to help keep your embryo safe. Let's look at the structure of an amniotic egg of a terrestrial organism, in this case, a chicken. We have an embryo which is developing within an egg, and that egg has a shell which is generally made up of a high percentage of calcium. Surrounding the embryo, we have extraembryonic membranes. These are membranes that further protect your embryo and also help to provide the embryo with certain life functions. Firstly, we have the chorion, and your chorion assists in gaseous exchange. We then have the yolk sac, and the yolk sac contains nutrients or yolk that the embryo can feed off of. Next, we have the Allantois, and the Allantois helps with gaseous exchange and also helps with excretion. So this is where the embryo will excrete its waste into. And lastly, with regards to your extraembryonic membranes, we have the amnion, which contains amniotic fluid, and your amniotic fluid helps to keep your temperature constant, and it also helps to prevent the embryo from experiencing a lot of shock if the egg has to move. So in other words, it plays a role in shock absorption. If we have a look at this image, we can see that the embryo is connected to all of these membranes. So I've highlighted the embryo in a dark green, and with a blue color, we can see that the embryo is attached to your yolk sac, it is attached to your allantois, and it is found within the amnion, and all of this is surrounded by the last embryonic membrane or extraembryonic membrane called the chorion. If we look at the place of development and how your embryo receives its nutrients, we come to the following terms on our slide: ovipary, ovovivipary, and vivipary. So the prefix ovi means egg, and the prefix vivi means life. And so ovipary, our very first one, looks at the development of your embryo within an egg, and we saw that in our previous slide. Vivipary looks at live birth taking place. So you have internal fertilization taking place. Your embryo develops within the mother. There is a placenta and an umbilical cord, which helps to provide your developing embryo with gases and also to remove gases from the embryo. And these structures, your placenta and your umbilical cord, also play a role in providing food to the baby as well. Ovovivipary is kind of like a combination between ovipary and vivipary, in which your offspring develop within an egg inside your mother, but there's no connection between the mother and the baby. An example of an animal which uses ovipary could be a chicken or a frog. An example of an organism which uses ovovivipary is a shark, and an example of an organism which uses vivipary is a human. One of the important processes that take place in this unit is the process of fertilization, and we remember that fertilization refers to the fusion of the haploid nucleus of your sperm cell with the haploid nucleus of your egg cell. And there are three phases that occur within fertilization. Firstly, the acrosomal reaction, then your cortical reaction, and lastly, the activation of the egg cell. Let us look at the first phase. The first phase which is our acrosomal reaction, starts when your sperm cell meets your egg cell, or your sperm contacts your egg. If we have a look at our sperm cell, we can see that here we have the sperm head. We will then have the sperm tail, which will move and allow it to contact the egg. If we then have a look at our egg cell, on the outer region we have a jelly coat. Then just below that we have a vitelline layer. Under the vitelline layer we have granules which contain calcium ions, and attached to the plasma membrane or the cell membrane of your egg cell are sperm binding receptors. So still in the acrosomal reaction, after the sperm contacts the egg, we see that your acrosome is then released, or rather your acrosome releases your hydrolytic enzymes. So your acrosome is the front part of your sperm head, and it contains enzymes called hydrolytic enzymes. And hydrolytic enzymes are responsible for breaking things down. And as we can see in this section, the hydrolytic enzymes are starting to break down your jelly layer within this region. The next stage of your acrosomal reaction occurs when your actin filaments of your acrosomal body or your acrosomal process bind to these sperm binding receptors. So here we can see our acrosomal process and our actin filaments, and they attach onto the sperm binding receptors. And that is shown nicely in this region, which I've now circled in purple. And what happens after this? We see that the plasma membrane of your sperm cell and your egg cell fuse. So there's a unification between the cell membrane of your gametes. And at this point, the haploid nucleus of your sperm cell will then be released into the cytoplasm of your egg cell. And this sets up a fast block to polyspermy. And what do we mean by that statement? Remember, there are millions and millions of sperm trying to enter into the egg, but we only need one haploid nucleus from a sperm cell. We don't need the nuclei of all of those other sperms. So once a haploid nucleus has entered into the cell, the egg cell will create a fast block to polyspermy. And it does this by ensuring that these sperm binding receptors fall apart so that no other sperm may release its nucleus into the egg cell. And that brings the first stage of fertilization, called the acrosomal reaction, to an end. Next we come to the second phase of fertilization, which is called the cortical reaction. Now the phases of fertilization don't happen in sequence. They happen at the same time. So we saw that your acrosomal reaction started when your sperm met the egg. And at the same time, your cortical reaction behind the scenes also starts. And the region in which your sperm meets the egg, those cortical granules, which contain those calcium ions, release those calcium ions. And that then forms a fertilization envelope. And this envelope also blocks the entry of other sperm cells from releasing their nuclei into the egg cell. But it's much slower, and so we say that this develops a slow block to polyspermy. So what does this look like on a diagram? Remember that this entire process that I've now highlighted in yellow is your acrosomal reaction. And as that is taking place, at the same time that all of this is happening, we see that in the region where your sperm is meeting the egg, your cortical granules start to release those calcium ions. Those calcium ions are released outwards towards the cell, and that then forms a fertilization envelope, which prevents more sperm from entering, as there's an envelope, a fertilization envelope, which prevents them from fully contacting the egg cell, and that is known as a block to polyspermy. It's much slower than the block to polyspermy setup in the acrosomal reaction. And then we come to our third and final phase of fertilization, called the activation of the egg. And it does not occur after the process of the cortical reaction. It occurs while the cortical reaction is taking place and finishes after the cortical reaction. So that's why we dedicate a separate phase to it. And as a result of the sharp increase in your calcium ion concentration, this increases the process of cellular respiration and the process of protein synthesis. We need to have energy in order to undergo a process from being a zygote to being an embryo, and that is why the process of cellular respiration is necessary. And the process of protein synthesis is also necessary because now we need to start producing proteins. This zygote is eventually going to develop into a fetus, and a fetus is filled with many, many proteins. And at this point, the egg is said to be activated, and that brings the process of fertilization to an end. So fertilization has three phases: the acrosomal reaction, the cortical reaction, and the activation of the egg. Once your egg is activated, it will undergo some mitotic divisions, and from this point we now consider that structure to be an embryo. And the next process we are going to look at is how this embryo undergoes its development. Next, we are going to look at the process of embryonic development. And this process includes three phases: cleavage, gastrulation, and organogenesis. Cleavage is a type of cell division that takes place without any growth taking place. So if we have a two-celled structure, under the process of cleavage, cell division takes place, and we will then form a four-celled structure. However, if we look at the growth or the size of this compared to its size after cleavage, more or less, obviously my diagram is not accurate, but more or less they are the same size. And that's what we mean when we say that cleavage takes place, in which there is cell division with little growth, little to no growth. If we have a look at it in terms of diagrams, we can see that as we move from a zygote to the resulting structure of cleavage called a blastula, your cell size remains the same. The result of cleavage is a blastula, and a blastula is made up of little building blocks called blastomeres, and in the later stage it forms a cavity, and that is called a blastocoel. Our next phase of embryonic development looks at gastrulation, and gastrulation is the rearrangement of the cells in your blastula, which can either be arranged into a 3 or into a 2-layer structure. If it's a 3-layer, it will be triploblastic, and if it's a 2-layer, it will be diploblastic. And a nice way to think about this is, for example, if you have a ball like a tennis ball and you press your thumbs inwards, that is, metaphorically speaking, what happens during the process of gastrulation. And once gastrulation has taken place, we then form a gastrula. There are 3 potential membranes. This includes the ectoderm, or the outer layer, the endoderm, or the inner layer, and in organisms that are triploblastic, we have the mesoderm, which is found between the ectoderm and your endoderm. If we had to look at the structure of a gastrula for a frog, we can see our layers, our endoderm, mesoderm, and ectoderm. We also find that if we think back to that tennis ball metaphor, that inner cavity that is formed is the archenteron, and your archenteron is like a very primitive early digestive system. We also have a yolk plug, a blastopore, and we have a blastocoel remnant. Our third phase of embryonic development looks at the process of organogenesis, which is the development of organs from your germ layers. Your notochord develops from your mesoderm, and your neural plate develops from your ectoderm. These are 2 examples, but there are many other examples of the organs that develop from your germ layers. In this diagram, we can see it nicely. Your ectoderm, which is the outer layer, undergoes some folds inwards and then becomes your neural tube. And on this slide, we can see all of the structures and organs that develop from each of your layers. For example, your ectoderm will give rise to tooth enamel, for example, and your endoderm will give rise to your digestive tract. We then come to our final part of this topic, and that looks at precocial and altricial development. And this development looks at the extent of yolk which has been used by the embryo at the time of birth or at the time of hatching. If an embryo uses a lot of yolk during its development, it can develop quite largely and to quite an extent, as so that when it's born, it is able to move around from birth, it is able to open its eyes from birth, it has fur from birth or from hatching, and that is called precocial development. Altricial development occurs when less yolk is used during embryonic development, and as a result of that, your offspring that are born or that hatch are often blind and don't contain feathers or fur. And when it comes to altricial young, there is a high extent of parental care compared to precocial young. So that brings our topic to an end. I hope that this was enjoyable and that you learned something. Thank you for watching, and I will see you soon.